Hi, everybody. I think we'll just start with some introductions and then perhaps a few others may join us. So my name is Professor Kristen Brown, and I am one of the leads for the MSc Regenerative Medicine.
Hello, I'm Professor John Connolly. I'm the other co-lead for the Masters in Regenerative Medicine.
Okay, great. So I just want to remind everyone that we are going to be recording this afternoon's session, and we'll be distributing to people who can't attend today. So if you're welcome to put your voice on and chat to us. But if you want to ask a question and aren't comfortable with that, if you just put it in the chat, if you type it in, we can address it as we go through. So what we would do today is I would start with a little overview of the course. And then Professor Connolly is going to talk about some of the research that we're carrying on at Queen Mary. And I think the most important thing is to give you the opportunity to ask us any questions that you have about the program. So we're going to try to keep our presentation short. We're happy to go back to anything if you want to go through in more detail, but really to give you the chance to interact with us and ask any questions that are on your mind. So I'm just going to share my screen and start off today. Okay, so we're here to talk about the MSc Regenerative Medicine. We're going to do a program overview and some research highlights. So that will be the format of our webinar, and then at the end we'll have a question and answer session. So what is regenerative medicine? Most of you, I'm sure, are very familiar with this. This is why you're here today. But for those of you who may be less familiar, it's the idea of using cellular biological components to regenerate damaged tissues or organs. And we think this is a really interesting field because it's interdisciplinary. It brings together experts in stem cell biology, biomaterials, tissue engineering, and clinical medicine. And to train students to work appropriately within this field, we need students to become familiar with concepts that cross these different specialties. So that's very much the aim of our master's program. So describing how the program is organized, we launched this program back in 2014, so we've been going for a while now. Our flagship program is a one-year full-time master's in regenerative medicine, but we have introduced some new streams, which I'll come to a little bit later in this talk. Something that we think is a real strength of the program is that it's interdisciplinary. It's collaboration delivered between the Faculty of Medicine and Dentistry, where Professor Connolly and I are based, and our colleagues in the School of Engineering and Materials Science. So we have modules that are led by both of these, the medical school and the engineers, and also projects that are offered in both medicine and in engineering. The foundation of the course is 8 taught modules. And then at the end of the master's, students undertake a dissertation research project. So if we just go through the course structure in a little bit more detail, starting the first term, which runs from September until December, students will take 4 modules. Each of these modules are 15 credits, so that makes up 60 credits in total. And the modules that students take are research skills and methodology. So this gives you a basic introduction to how we do research and some of the main techniques and how to give you an oral presentation, write a good paper, all that kind of stuff. And this module is led by Dr. Daniele Bergamaschi and Dr. Shuenong Bo. The second module is Molecular and Cellular Basis of Regeneration, led by Professor Cleo Bishop. We have a module in Stem Cell and Developmental Biology, which is led by myself and Dr. Miguel Branco. And then a module entitled Advanced Tissue Engineering and Regenerative Medicine, and that's led by Dr. Tina Chowdhury. So this is the standard 4 modules that are taken by the majority of our students. The only exception is if students have studied at Queen Mary before and have taken one of these modules before, then we have some alternatives, but I don't think that applies to anyone in this room today. Feel free to drop us a note in the chat if it does. So then we get to semester 2, which starts just after your January exam. So in January, we examine semester 1, and then we start semester 2 at the end of January, and that runs through April. And during this term 2, students can choose 4 out of 5 modules. Again, each of the modules are 15 credits. So in total, the students take 60 credits. And the students can choose between medical ethics and regulatory affairs, which is led by Professor David Lee and Professor Liz Tanner. Biomaterials and regenerative medicine, which is led by should be Professor John Connolly. Tissue-specific stem cells, which is led by Dr. Matthew Cayley. Induced pluripotent stem cells and genome engineering, which is led by Dr. Jürgen Groot and Young Yao Lin. and neurodegenerative diseases, which is led by Dr. Sarah Morgan and Dr. Agnes Nishimura. So you can see this last module is more focused on neuroscience, and we have a number of modules that we share with students on our MSc Neuroscience. So our students get to know not only students studying regenerative medicine, but also students studying neuroscience really well. And then during term 3, starting in May, our students undertake their final research project. And the research project is worth 60 credits. So it's 4 times as much as at each of the taught modules. And the students join one of our research labs to undertake a 12-week long research project. And how we do this is the project titles or topics are distributed in October, and the students have the chance to read these project descriptions and also meet with the supervisors if they wish to during term 1, which we really encourage. And then students submit a preference list of basically ranking their top choice projects. And then we just try to assign as many students to one of their top choice projects as we can. And the project titles are disseminated early in term 2. Students then begin working in the lab in May, and their dissertation and their oral presentation at the end of the projects are due in late August. So it really is a full year course going from September through to the end of August. I just want to give you some examples of research projects that are actually being offered to our current students. So you can see these top 2 projects are focused more on stem cells. So the first one is related to epigenetic regulation of transposons and trophoblast stem cells. So looking really early during development. And then at the other end of the spectrum, we have a project focusing on the impact of aging on induced pluripotent stem cell potential. We also have projects that are more engineering focused. So we have granular hydrogels for 3-dimensional printing of vascularized human tissues, which is being offered, and ECM production and next generation bioreactors for tissue engineering. So we have a really nice blend of projects that are kind of more biology focused, more stem cell biology focused, but also those that are more on the bioengineering side of things and biomaterials side of things. You can see on the right-hand side, I have a couple of nice images here, which I stole out of some of our previous students' theses. We always get praised on the quality of the projects that we offer and also the data that's generated by our students. And to generate this data, our students are able to use the very cutting edge equipment that all of the PhD and postdoctoral researchers in our institutes have access to as well. I just want to talk a little bit about teaching plans. Our teaching and learning will continue to be delivered by mixed mode education. So what this means is that the majority of the teaching is delivered through on-campus activities. All of our lectures, we have a lot of discussions, we have student presentations, will be delivered on campus. So we've moved away from obviously the emergency response that was during COVID, and everything's available in person. But we do support that with some asynchronous online activities. So a reading that students can undertake in advance of their teaching, videos to support the teaching, sometimes online quizzes. So we have a mix of both on-campus activities and online activities. Students are encouraged to come onto campus whenever possible, but if for some reason they can't come to campus, they have the opportunity to log on to taught sessions remotely. So this is why it's called mixed mode education. So if you're feeling a bit unwell one day or just can't get to campus because of transport, there's the option to log on online. But I do really want to emphasize this is a very interactive course, and the best experience is by being on campus with your classmates, asking questions and interacting with their teachers. Finally, our exams will be delivered online. So the students have been this year taking exams that typically last 4 hours, and they can do that in the comfort of their own home or whatever environment they choose to sit the papers. This year, we are expanding our portfolio of programs. We're really excited about this because we want to continue to be a very inclusive course, and we want to be able to take students who maybe can't commit to a full-time program. So we have added a part-time MSc Regenerative Medicine. This degree will be completed over 2 academic years. I don't think anyone in this meeting has applied for the part-time program, but if you have, feel free to pop questions in the chat and I'll go into more detail about that program. And the other option that we've made available is a PGDip, a postgraduate diploma, and we are offering this both full-time and part-time. So for the PGDip, students complete the 8 taught modules, either over 1 or 2 academic years, depending on whether they're full-time or part-time. But there is no final research project. But the good thing is graduates with the PGDip can apply to complete the master's within the next 5 years. So if a student decides they want to come back and do that final project, they can apply to return to Queen Mary and undertake that final research project. So we hope that this will give our students, our potential students, more flexibility in how they engage with the program. What do our students do after the master's? Our students, their outcomes have been really excellent. We have a large number of students now who've gone on to PhD studies, both within the UK, but also internationally. We have students who are working in research, either as research assistants in academia or in industry, whether that's large industry or smaller startups. We have a number of students who've gone on to medical school and the students who've gone into different career paths like science publishing or teaching or consultancy. And on the right-hand side here, we have a picture of some of our graduates. Graduation is around Christmas time. I just wanted to highlight one student in particular, Jengis. So he graduated with his PhD, went on to and is now running a stem cell laboratory in Maastricht. And he comes back and he lectures on the program every year about his experience that he's gained since he completed his master's with us. We also have a LinkedIn alumni network. So all of our graduates can be linked together and can advise each other of opportunities and help each other if they have questions about different career paths. And they're supported by Queen Mary's Career and Enterprise team. Not only are they supported while they're with us, but also for 3 years after graduation. So we really want to make sure that our graduates have great opportunities to get started earlier in their career. And finally, I just want to say we look forward to you joining the program. If you have any questions, please either pop them in the chat or hold them. And then when Professor Connolly finishes his talk, we will have a lot of time to take your questions and interact with you.
Great. I'll just go ahead and share my screen. Right. Can you see that all right?
Yeah, it's perfect.
Okay. So what I'd like to do today is to highlight some of the really exciting and excellent research that's going on in regenerative medicine here at Queen Mary, and hopefully give everyone on this meeting a better idea of what's going on and get you excited about regenerative medicine. So first off, just to kind of introduce the research environment at Queen Mary. So we are a member of the Russell Group, which are research-intensive universities in the UK. And we have particular strengths in medicine and dentistry, as well as science and engineering. And so both of these come together for this program in regenerative medicine. And supporting this excellent research are outstanding facilities and infrastructure. So the laboratories are state-of-the-art, and the students will have access to this through their research projects. And then we also have two outstanding hospitals. So St. Bartholomew's Hospital, which is actually celebrating its 900th anniversary this year, which is pretty amazing. So a very long and rich history of medicine. And then the modern Royal London Hospital, which you can see down the bottom in blue here. And so these are the two teaching hospitals, and many of the clinical academics that lecture on the program practice within these research hospitals. And so then we think about regenerative medicine research that's going on at Queen Mary. It is very diverse and covers a lot of different topics. And so I've highlighted here just a few of those. So we have some major programs in cardiovascular regeneration. And so this uses things such as stem cells derived from bone marrow, engineered cardiac patches, the use of exosomes as a therapeutic, and reprogramming of cells for therapeutics. There's also a number of different research streams related to connective tissue repair. So we have a big center for skin research and wound healing. And then there are a number of other researchers interested in bone and joint health, regeneration following trauma, and regenerative dentistry. And then in addition to this, we have a strong interest in cancer stem cells. So these are the stem cell or population of cancer cells that are responsible for tumor initiation and recurrence. And so this is a big area of research and supported by the Bart's Cancer Institute. And then there's also a number of different groups interested in rare diseases and understanding their pathogenesis and developing new strategies for therapy. And these take approaches including genome engineering, gene therapy, but also stem cell-based models to better understand the pathogenesis of these diseases. So this is kind of a broad overview of regenerative medicine research here at Queen Mary. And so what I'd like to do as well is to focus on a couple particular areas of strength and interest for us. And one of those is the development of next generation models of human tissues and diseases. And here the concept is that we want to move away from our standard research tools, which include either cell culture, which is overly simplistic and doesn't accurately replicate human biology, or animal models, which have ethical implications and likewise don't always replicate human biology. And to try to use some of our engineering tools to recreate tissues and organs in the lab. And these include things such as organ-on-chip technology, stem cell-derived organoids, and 3D bioprinting. And these strategies try to build more physiologic models, including the three-dimensional structure, having multiple different cells within the models, and importantly, using human cells with the right physiologic cues, such as oxygen and mechanical forces. And so just to give you one example of some of the infrastructure that helps support that is the Create biofabrication, which is based here in the Blizzard Institute. And so this is a new core facility that we launched in 2018, and it provides researchers within the university access to state-of-the-art equipment for 3D bioprinting and the fabrication of microfluidic devices, which you can see an example of here on the right. And so we have a wide number of users, and it's supported by a full-time facility manager. And importantly, students on the course will carry out laboratory practicals within the facility to gain some experience in these techniques. And then we also support a number of different thesis projects related to bioprinting and organ-on-chip technologies. This is further supported by an additional center, which is the QM and Emulate Organ-on-Chip Center in the School of Engineering and Materials Science. And this provides access to the Emulate Organ-on-Chip platform, which is a fully optimized and commercial system. And then finally, to highlight one specific example of how we're using stem cell-based models and 3D tissue models to advance our understanding of diseases and human health, I wanted to highlight some of the work of Dr. Young-Yao Lin. So Dr. Lin is one of the lecturers and module leads for the genome engineering module, and he's hosted a number of MSc project students in his lab. And one of his main research interests is developing new therapeutic targets for Duchenne's muscular dystrophy, or DMD. And to do this, Dr. Lin's group takes induced pluripotent stem cells derived from patients with DMD, and then uses CRISPR-Cas9 to correct them. And they can see that the corrected cells regain some key features of skeletal muscle, which are lost in the disease. They also fabricate 3D mini muscles. So here you can see this mini muscle that they replicated in vitro, which forms this nice striation pattern characteristic of skeletal muscle. And then importantly, they've used these tools to discover new biological pathways and therapeutic targets. And so, for example, here's a graph from one of their recent papers where they showed that by treating the DMD cultures with a specific compound, they can improve the strength of muscle contraction and improve functionality. So I think this is a really nice project because it highlights kind of many of the aspects of regenerative medicine, from basic stem cell biology and genome engineering to tissue engineering, and taking that through to the identification of new therapeutic targets that will ultimately benefit patients with this disease. So just to kind of briefly summarize, I think hopefully you can appreciate that There's a very rich environment for interdisciplinary and regenerative medicine research here at Queen Mary. And we have particular strengths in tissue and disease modeling, both from the stem cell biology side, but also on the tissue engineering and fabrication side. And importantly for you guys, researchers who are active in these areas will be involved in the instruction on this program, but also supporting the hands-on research training through the thesis projects. So that's all I wanted to introduce today. So I'm happy to take some questions. So I'll stop sharing. We can go back to the main screen. So if people have questions that they'd like to ask, feel free to raise your hand or put them in the chat. I think.
Actually, just to get the ball rolling and to make sure the chat's working well, would you mind typing in where you are today? That'd be quite interesting for us. What city you're in or what country? All right, so we've got a nice variety here.
Some strong representation from Sheffield.
Go Sheffield. Excellent. I recognize one name on the list. Welcome to the person that I know. All right, so we've got a question. We'll go back up to the top.
Oh, good. We've got some questions now.
Great.
Should I take this first one? So Mariana asks, do the dissertations of the students have the chance to be published in the future? Absolutely. So we do intend that the students on their projects are doing, real research. They're not replicating someone else's work. They're doing new research. That's saying that it's unlikely that the thesis research would be published as a paper on its own, given the time constraints. It's more likely that the thesis research could become part of a larger paper or study. And certainly that's happened a number of times on the program where pieces of data from a student's master's thesis go into a larger publication.
Good. I see we have a question from Emily. It says, I've applied for the part-time course. Could you please run through how this will work, particularly the research project? Well, first of all, Emily, thank you for applying. That's exciting because we've just gotten this launched and are very something we've been talking about doing for a while and are really pleased. So how it will work is, as I mentioned in the presentation, normally students take 4 modules at a time in the first semester and the second semester, and that makes up 8 total modules. For the part-time program, students will take 2 modules in the first year in the first term and 2 in the second term. And then the second year, the same, 2 and 2. So that will make up the 8 modules. And the research project, we built in a little bit of flexibility. So we have in the paperwork said it will be delivered during the year 2, semesters 2 and 3. So we can kind of work with our part-time students as to what would work best for them, whether it's better to spread it across semesters 2 and 3, or perhaps take the summer off like the full-time students do and really just focus on the project, full-time after all the taught modules are finished. I hope that's clear, but feel free to ask any follow-up questions that you may have. But generally, you would be, as part-time students, sitting the modules at the same time as the full-time students. We think it's really important so that we don't expect a huge number of part-time students, so they feel part of the larger group. Do you want to do the next one?
Sure. So Ajawara asks or wants to know if the coursework due dates overlap. So there will be some overlap in terms of having assignments on the go, but we do try to separate the due dates a little bit so the students don't have, say, 3 pieces of coursework due all on the same day, but towards the end of the semester when modules are wrapping up, there might be several pieces of coursework due on different modules, but it could be that there's one due 1 week, one due second week, and one due kind of later on in the second week. But we do try to spread them out at least by a few days.
Okay. We have a student who says, I may not be related to this meeting, but I got an offer, but the admissions, you believe you shouldn't be an international student? That's absolutely fine. Could you send us an email afterwards, and we will work with admissions to make sure that your status is correct. Our email address is on the course website, or otherwise just look up either myself or Professor Connolly, and we'll sort that out for you. All right, any other questions?
I'm going to take this one. So Mariana says, as a medical student, I have no background in engineering. When discussing with past students, they told me that I'll have no disadvantage. Is that right? Yes, that's completely correct. So we have students from all sorts of different backgrounds, and know that people are coming in from with different sets of expertise. And so you don't need any formal engineering training to be able to complete the engineering modules. We also offer a kind of introductory session to some of the key concepts in engineering that you will then need to know for those modules. But in the past, students from all backgrounds, in particular the medical students, do very well on the tissue engineering modules. So I wouldn't be concerned about that at all. I would say it might seem a little bit strange or foreign early on because the terminology is different. Some of the concepts are a bit different. So there is a bit of a learning curve, but in the end, the students from all backgrounds do just as well as students with engineering backgrounds.
Can we get assistantships under professors to work in their labs during the course? That's a really good question. We get that question quite frequently. We don't arrange for students to be working in our colleagues' labs during the course. So there are a couple reasons for this. It's a master's for people who usually really want to carry on careers in research, want to go on and do things like PhDs or medical school or research careers. So it's a challenging course, and we think it's best for students to really be focusing on the taught content during the year. And then when they get to their final research project, they're very well prepared for how to analyze data, how to present data. Having said that, if students want to try to self-arrange opportunities, they're very welcome to do so, but it isn't something that we facilitate as a program. And based on our previous students' feedback, I think most of them kind of understand once they get through the year that it was better for them to focus on their studies and then get that research experience at the end. What we have had students do in the past, we can't promise this, but we have had students who've completed the master's and then sometimes stay on a bit longer to get some additional experience if they make a local arrangement with their supervisor. So that probably would be a better way to gain some additional experience in my opinion. Do you want anything to that answer?
No, I think that's good. I think there was one previous question about from Isha, who asked, she would like to know if they get the opportunity to interact with the teaching hospitals throughout the course. So that's a really good question. I think probably the main way that students will interact with the clinical side is through lectures from clinically active faculty who can give that perspective on what's going on in the hospital, how do you translate technologies to patients, and the implications of that. Students probably won't spend any significant time actually in the hospital or in that setting. Most of it will be in the laboratories, but that may depend on the nature of the project, if they're collecting tissue samples or something, but that's most of the students who are in their research projects are based within the laboratories. I think there have been a few instances where clinically trained students, so students who have completed a medical degree previously, have reached out and done some shadowing or made some connections with other clinicians in the hospital, but that's been self-organized. But the main interaction with the clinical side of regenerative medicine will be through the lecturers and clinical academics. Looks like there's another question from Emily.
Yeah, for part-time, what would be the recommended study hours per week for the course? That's a good question, Emily. I've just offered to have a chat with you privately, because I think you're the only one on this call who's applied for part-time, and it probably would be good for us to go through a little bit more detail. But I think for guidance for everyone on the call, for those of you on the full-time course, we would consider it a full-time, commitment, much like a full-time job. So part-time, I would kind of predict that it would be about half of that a week. So, maybe 20 hours a week or so, because you'd be in half of the number of modules at any particular time. The only exception to that might be during the final dissertation project, when students, to get the most out of the project, they work quite hard. And even for part-time, that will be more than kind of half time during that period. Like everything in life, the students who put the most in, especially in the research part of the project, I think get the most out of it.
There's another question about clinical training. Yeah, I can follow up on that. So just to be clear, this program is a science-based course, and we do not provide clinical training on this program. We have lectures from clinical academics who have that experience and can provide insight into translation, but we don't provide actual clinical training. We do have some students on the course who come from medical or clinical backgrounds, and they sometimes self-organize interactions or outside training with the hospital, but that's all on their own. So just to be clear, this program is science-based rather than clinically based. I think there are questions about email addresses. I'll just put my email address in the
David's done it already. I was just going to do that.
Oh.
Thanks, David.
David's ahead of us. Yes, excellent.
I just want to go back. How will we be trained before our project, or will we be trained in the lab? So that's a really good question. We do have a little bit of lab experience during the course, but most of it is not in the lab. I want to be clear about that. So for example, we tend to have a practical on qPCR. So the students do a little bit of work in the lab, but actually it's more about using the online tools and how you analyze, and set up your primers and analyze the data. And the reason that so most of the training is in the lab during that final 12 weeks. And the reason is that it's just, you learn so much more, even in their first week in the lab, because you have one-to-one mentorship usually with a PhD student or postdoc, or perhaps the lab head. You're using the real equipment, not kind of the, those of you who just come out of undergrads, the kind of equipment that you get, when you're in undergraduate programs, you're using the state-of-the-art equipment. So we find that our time is best spent before the final projects, really working with students, developing their communication skills. So how do you analyze scientific data? How do you communicate it? How do you give a journal club? How do you write a dissertation? And we leave most of the lab work to that final project, where you can get that one-to-one, really bespoke training.
Yeah, so there's another question about practical training with iPSCs. So I guess along those same lines, we don't have any kind of broad laboratory practicals for iPSCs, but leave that to the thesis projects. So students who are doing a project that uses iPSCs will get very extensive experience culturing iPSCs and working with them really on a daily basis. So in that sense, training on iPSCs will depend on what you choose for your thesis project. So if that's an area of interest, then I would recommend focusing on those projects. And maybe just to add on to the lab practicals, we do have one afternoon session on the biomaterials module where the students are in the lab and get some training and experience on the 3D bioprinter and making organ chip devices. But that's mostly just a demonstration, and the students is one afternoon. But it helps give people a feel of what those systems look like and what those techniques are like, because they are quite different from the other cell biology methods. Great.
Okay, any other questions or things you'd like to know about, or things you'd like us to cover in our next webinar? We will be doing one or two more as we approach the academic year. I think we've covered most things.
Yeah, I think we've covered quite a bit. Oh, there is another question about jobs.
Do you want to start on that one?
Sure. So yeah, students who graduate from the regenerative medicine program go on to a quite diverse range of careers. Probably the most common is going on to a PhD. So I think we have maybe about 30%, is that right? Go on to do a PhD at some point. We do have a number of medical students and clinically trained professionals on the program, and they typically go back to their clinical jobs. And then aside from that, there's a pretty wide range of careers. So some students will go on to research jobs in academia or industry, particularly like pharmaceutical or biotech companies. And then we've also had a number of students who go into kind of public policy and grant funding agencies or science publishing. And I think a few people have even gone on to teaching roles. So it's varied, but probably the most common route after graduation is going to a PhD. And that might not happen immediately. Some students work for a little bit as research assistants in a lab and then move on and do their PhD.
Yeah, and I think something that we've developed, as you said, we launched the program in 2014. We now have a nice network of PhD students around the world. So we have PhD students in North America, in Asia, in several places in Europe, a lot of institutes in the UK. And I think that's been very helpful for us, understanding some good places to recommend that students look into. And also we have this LinkedIn group, and our graduates are now starting to share opportunities at the places that they work with you guys, and I think that's really building up to a nice network as we progress. I think something that's really important is that some people get to the lab and they realize they don't want to do lab research. So we try to build up what we call transferable skills, which I'm sure is a phrase you've heard a lot, but really the confidence, it's a small program. So students do give a lot of talks and interact a lot and do group projects. And at the end of the program, they always comment on how much their presentation skills have improved, which I think is beneficial for really any career path that they choose to pursue. And we're always amazed when we see the final dissertation presentations, how much people have improved in the kind of year that they've been with us.
So there's a question about PhD opportunities. So there are a number of different routes to finding a PhD studentship. So those can be within the UK or in any other country. And so we've had students who have gone on to PhDs here in the UK and here at Queen Mary, but also in the US and Europe and in Asia. So all over the place. And those PhD studentships are advertised kind of throughout the year, and we can help direct students to those sites and how to search for those. So hopefully that answers the question, but I think it's more kind of one-to-one guidance that your academic advisor would provide.
Yeah, that's something we haven't mentioned, actually. So each student on the program has an academic advisor, and usually we look after maybe 3 to 4 students on this program. So we get to know our advisees very well, and they get to know us very well. So there's that person who, will be at your side throughout the year, and even after you finish the program, they'll get to know you very well, which helps them to write letters of reference in the future. But they'll also begin to understand, your career path that you want to pursue, or any concerns that you have, and they all are very familiar with the university support services. There's a lot of support available, for example, for students with different disabilities, or who require some support through advice and counseling. And sometimes, you don't want to talk to everyone about that, but having that one person who knows you well can be really helpful. I see a question about wanting to know more about modules and the coursework. So I'll break that down a little bit and then see if my colleague wants to add anything. So the modules are 12 weeks long, and we tend to teach typically 11 of those weeks. So usually halfway through, we have a little bit of a break for a week. They are, we have about 3 hours of talk time each week. And in a typical session, usually we have a couple of lectures, and then in the afternoon, something a little bit more interactive. So sometimes it's student presentations or some group work, so usually that's kind of the layout of a particular day. In terms of coursework, we try to have a variety of coursework and try to develop the skills that you'll need as you go on the program. So you may be doing a literature review, you may be doing a mock grant proposal, which Professor Connolly uses on his module. You may be doing a journal club presentation where either a paper is chosen for you by us, or as you progress in the course, you and your colleagues choose a journal club paper and then present it to the group. So we try to really use a variety as the year goes on to make sure that that things get interesting, but also that you develop a different set of skills. And I think it's really important because not everyone, of course, there are some exams as well, but not everyone, enjoys or is good at exams. And actually, the final piece of work, of course, your dissertation really is just a massive coursework. How do you present figures, and how do you write up a dissertation? So it's building up those skills with smaller pieces of work as the year goes on. Is there anything you want to add to that?
No. Yeah, I mean, I guess maybe just to say that most modules have 2 pieces of assessment, usually one exam and one piece of coursework. Some have 2 smaller pieces of coursework, but yeah, like you said, it is varied.
One thing I'll add is, I know there's a few medical students on here. It is quite a change from studying medicine, because there are, and I think sometimes the medical students are surprised when they come to do a master's. There's definitely more emphasis on coursework. Because it's a smaller program, you have the chance to give oral presentations and listen to each other present. So it's not us, it's also us learning from you guys, not just you learning from us. So that's, I think, one of the joys of teaching and learning at postgraduate level. But it is a change from medicine, where often there's a lot of weight on just the end of year exams. So our module exams are spread throughout the year. So we do them in January and then at the end of semester 2. So that really students over the summer period can usually just focus on their research project. Okay, I think we'll give you another minute or so to see if there's any final burning questions. Otherwise, this recording will be posted or advertised to you by our colleague who is running this session, and we will be organizing some additional webinars going forward. Also, this is a small program, so if any of you have any questions that you're not comfortable asking in this forum, if you use the email addresses that are in the chat, either Professor Connolly or I will be very happy to arrange an individual Zoom call with you and address any concerns that you have. So that's absolutely no problem. All right, last chance. Thank you, David. All right, happy to close.
Yep. Thanks, everyone. It was nice hearing from you, and glad you were able to log in and join us today.